For all things health and fitness, this is the Army Gymnastics Podcast.
The Army Gymnastics Podcast.
Discover a fitter, healthier you at armygymnastics.com.
Welcome to The Deep Dive, the show where we take a stack of your sources, articles, research, our own notes, and extract the most important nuggets of knowledge or insight, giving you a shortcut to being truly well-informed. Today, we're embarking on a crucial journey, one that touches so many lives.
Yeah.
It's our deep dive into Down syndrome and Alzheimer's, uncovering the hidden link.
Yeah, it's a really important topic.
It really is. Alzheimer's is a disease that, strips away memories, connections, identity.
Yeah.
It leaves just a profound and often heartbreaking impact, not just on the individuals, but their entire families too.
Absolutely devastating.
And this burden, for many, feels tragically amplified for individuals with Down syndrome. The symptoms of Alzheimer's can manifest shockingly early, sometimes decades before we see it in the general population.
Right. It's a much accelerated timeline.
It's an incredibly challenging reality.
Yeah.
But, amidst this, there's just immense dedication from researchers across the globe, a relentless pursuit to understand and hopefully alleviate the suffering. Their work brings a powerful sense of hope, I think, that new discoveries can and will bring us closer to effective treatments and maybe even prevention down the line.
Yeah, and that's really what we want to focus on today, isn't it? The science that's driving that hope.
Exactly. Our mission today is to unpack the very latest scientific breakthroughs that offer, well, a new lens through which to view this complex and often heartbreaking connection. We're going to reveal some surprising culprits and explore potential new avenues for treatment that are emerging from this cutting-edge science.
Okay.
By the end of this deep dive, you'll not only understand the what, but also the why, and crucially, the what's next in this critical area of research.
It's absolutely true. The emotional weight of Alzheimer's, particularly in the context of Down syndrome, is just immense. But like you said, the scientific community is making remarkable strides. It's genuinely exciting to share these insights.
Good.
For this deep dive, we've looked at a range of recent research articles, some groundbreaking studies, actually, from places like USC, published just recently, June 2025.
Wow. Really current stuff.
Yeah, very current. And also comprehensive reports from leading neuroscience foundations that are really at the forefront here. So we're going to delve into the very cellular mechanisms that contribute to this accelerated progression of Alzheimer's in Down syndrome.
Okay.
And just as importantly, we'll look at the promising therapeutic paths that are now emerging from this deeper understanding.
Okay, let's unpack this foundational connection then. We know there's a strong, undeniable link between Down syndrome and Alzheimer's.
Right.
But how exactly are they connected on a biological, maybe even a genetic level?
Right. So it all starts with the genetics of Down syndrome itself.
Okay.
Individuals with Down syndrome have a third copy of chromosome 21 instead of the usual 2. That condition is known as trisomy 21.
Trisomy 21.
Got it. Now, here's where it gets really critical for our discussion today. Located on that very chromosome, chromosome 21, there's a specific gene. It's called the amyloid precursor protein gene, or APP gene for short.
APP gene. Okay, I've heard of that one.
Yeah, you probably have. This gene is fundamentally involved in producing certain proteins, specifically one called amyloid beta, or A.
Amyloid beta, the plaques, right?
Exactly. These A proteins are kind of notorious in Alzheimer's research because when they misfold and clump together, they become the building blocks for those characteristic sticky plaques that accumulate in the brains of people with Alzheimer's.
Like microscopic gunk clogging up the works.
That's a good way to think about it, yeah. So the direct and really profound consequence of having this extra third copy of chromosome 21 is that individuals with Down syndrome produce about 50% more APP protein throughout their entire lives, just because of that extra gene copy.
50% more. Wow.
Yeah, it's like their body is constantly running with an overdrive switch flipped on for this particular protein production line.
Okay, I see.
And this increased production, well, it significantly heightens their risk of developing Alzheimer's disease.
Right. And the statistics you mentioned earlier are pretty stark.
They really are. By age 60, roughly about 50% of all people with Down syndrome show clinical signs of Alzheimer's disease.
Half by age 60.
Yeah. And to put that into perspective, that's approximately 2 decades, 20 years earlier than what we typically see in the general population.
2 decades. That's a massive difference.
Yeah.
It really tells you there's something unique and accelerated happening there.
Absolutely. And this accelerated timeline in Down syndrome, it's not just, a tragic statistic for those families, though it certainly is that. It's actually critical for broader Alzheimer's research worldwide.
How so?
Well, think of it as a unique sort of natural experiment, or maybe like a fast-forward model of the disease.
Okay.
By studying why Alzheimer's manifests so much earlier and often more aggressively in individuals with Down syndrome, scientists get this invaluable opportunity. They can examine the very earliest stages and the fundamental drivers of the disease in a way that's much harder otherwise.
Right, because in the general population, it takes so long to develop.
Exactly. For most people, Alzheimer's development can span many decades. It makes it incredibly challenging to observe those initial subtle changes, the molecular triggers that start everything off. But in the Down syndrome population, because of that extra APP gene, these processes are essentially sped up. Researchers can observe them more clearly, more efficiently.
So it gives clues that could help everyone, potentially.
Precisely. It provides crucial clues that might be applied to understanding and treating Alzheimer's in the general population, too. Insights into fundamental disease mechanisms that, might otherwise take decades or even longer to uncover in typical aging studies. It's a unique window.
A unique window. Okay, so here's where it gets really interesting, you said, this recent research that's uncovered a major clue about these unique brain changes.
Yes. This is building on that foundation. New findings coming out of USC, published, like I said, June 19th, 2025, they revealed something quite astounding.
Okay, what is it?
People with Down syndrome who also have Alzheimer's, the condition often called DSAD, for Down syndrome-associated Alzheimer's disease, they have twice as much iron in their brains.
Twice as much iron. Compared to who?
Compared to individuals with Alzheimer's alone, without Down syndrome, and also compared to healthy controls, double the amount.
Double. Wow. And is this iron just everywhere or in specific spots?
That's a key point. It doesn't just accumulate randomly. This excess iron concentrates in very specific critical areas, precisely where brain cells are actively processing those amyloid proteins we just talked about.
So right where the amyloid action is happening.
Exactly. It seems to be right where the trouble begins, almost like it's setting the stage for more damage in those vulnerable regions.
Okay, so the next obvious question then is, where is this extra iron coming from? How does it get there?
Yeah, good question. Researchers suspect the source is tied to microscopic leaks in brain blood vessels. They're often called microbleeds.
Microbleeds. Like tiny hemorrhages.
Sort of, yeah. Tiny, often invisible leaks. And these are known to be far more common in Down syndrome patients.
Why is that?
Well, it's believed that the very same elevated APP protein, the one that creates amyloid plaques, might also make these tiny blood vessels more fragile and prone to leaking.
Ah, so the APP gene strikes again, basically.
It seems that way. So you can imagine these tiny internal spills happening within the brain where iron, which is carried in the blood, seeps directly into the brain tissue.
And then it builds up in those specific areas.
Right, creating this problematic, potentially toxic buildup. It draws a direct line from the underlying genetic predisposition, that extra APP gene, to this observed pathology of excess iron.
Okay, so we have this excess iron concentrating in key brain areas. That's the what. Now, you mentioned the how. How does this iron actually cause damage?
Right. So the research indicates that this excess iron isn't just sitting there passively. It actively triggers highly destructive chemical reactions within the brain cells.
What kind of reaction?
These reactions lead to what's called oxidative damage or oxidative stress. And it specifically targets and damages the cell membranes.
The outer layer of the cell.
Exactly, which are primarily made of fatty compounds called lipids. So imagine like an internal rust process fueled by this excess iron, specifically attacking the delicate protective skin of the brain cells.
Rusting from the inside out. That sounds bad.
It is. And this isn't just any kind of random cell death. The studies point to a relatively recently discovered and very specific type of cell death called ferroptosis.
Ferroptosis. Okay, break that down.
Sure. The name itself gives you a clue. Ferro for iron and ptosis for falling or death. It's essentially iron-driven lipid peroxidation.
Lipid peroxidation. Destruction of those fatty membranes.
Yes, destruction of those vital lipid membranes by oxidation, and it's all fueled by this problematic iron. This mechanism, ferroptosis, is now identified as a really key driver behind the accelerated dementia we see in this vulnerable population.
Wow. Okay, that's a major piece of the puzzle.
It really is. And what's particularly concerning, what really amplifies the problem, is that these DSAD brains also show evidence of weakened antioxidant defense systems.
You mean they can't fight back as well?
Precisely. Normally, our bodies have these incredibly sophisticated protective enzymes. Think of them like tiny repair crews or firefighters that step in to repair this kind of oxidative damage.
Right.
But in DSAD brains, the activity of several crucial protective enzymes is found to be significantly reduced.
Oh, it's a double hit.
Yeah,
More damage happening and less ability to fix it.
exactly. It's a devastating combination. More rust forming, and the rust removers aren't working properly.
Okay. And you mentioned something about lipid rafts
Yeah.
being hotspots.
Yes. If we zoom in even further on the cell membrane, the research highlighted these lipid rafts.
Yeah.
You can think of them as specialized control centers or processing hubs on the surface of brain cells.
Okay.
They're tiny platforms, crucial for cell communication, and really importantly here, for how proteins like APP are processed.
The protein that starts the amyloid cascade.
The very one. Now, in DSAD brains, these critical hubs weren't just damaged. They showed 3 times more oxidative damage compared to healthy controls.
3 times.
3 times. And critically, they also had significantly fewer of those protective enzymes we just talked about. So their command centers were essentially under heavy attack.
Making them weaker.
Weaker, and maybe more prone to making errors that accelerate the disease. And to make matters even worse, these damaged lipid rafts also showed increased activity of an enzyme called beta-secretase.
Beta-secretase. What does that do?
That enzyme is involved in the very first step of cutting the APP protein. It's one of the molecular scissors that snips APP in a way that leads to the production of those problematic ACAP proteins.
Oh, wow. So the damaged areas are actually producing more of the bad stuff.
It appears so. You have this perfect storm scenario. Increased damage in these critical processing centers, combined with increased ACE production right there, it inevitably accelerates the growth and spread of amyloid plaques throughout the brain.
So what does this all mean for the individual's brain cells then? It sounds like they're facing this truly devastating double threat, as you called it.
That's exactly right. On one hand, they're experiencing significantly more iron-driven damage, that destructive oxidative stress, the ferroptosis. And on the other hand, their natural defenses, their antioxidant systems, are simultaneously weakened. They're profoundly less able to fight back or repair themselves from this onslaught.
It really paints a clear picture of why the Alzheimer's progression is so accelerated. It's a direct pathway to widespread brain cell death.
It is. And tragically, that means earlier and more severe Alzheimer's symptoms. It reveals the unique vulnerabilities at play in a much more detailed and, frankly, more alarming way.
Okay. Now, you also mentioned another specific discovery related to those weakened antioxidant defenses, something about GCLM.
Yes, exactly. Building on that understanding, researchers identified another crucial and perhaps often overlooked vulnerability. They focused on an enzyme called GCLM.
GCLM, what's its role?
GCLM is absolutely essential because it's involved in producing glutathione.
Glutathione, I've heard that term. It's an antioxidant, right?
It is. You can think of glutathione as your brain's primary internal bodyguard against cellular damage. Arguably, it's the brain's most important antioxidant, like the master detoxifier and protector molecule.
Okay, so very important. What did they find about GCLM and DSAD?
The finding was quite alarming. GCLM activity decreased by a significant 60% in DSAD patients compared to healthy controls.
60% decrease. That's huge.
It's a massive drop. This isn't just a number. It reveals a fundamental Achilles heel in the DS brain's defense system.
So the brain isn't just facing more damage from the iron.
Exactly. It's also severely compromised in its ability to manufacture its very own most important protective molecules. Less glutathione means less ability to neutralize those damaging reactions.
Right.
This profound reduction in the brain's capacity to produce glutathione provides critical insight into why maybe standard Alzheimer's treatments haven't been as effective in Down syndrome patients.
Because those treatments might not address this specific iron glutathione vulnerability.
Potentially, yes. They might target amyloid or other pathways, but if this core antioxidant system is so depleted, the treatments might not gain as much traction. It really highlights the urgent need for targeted approaches that consider this unique biological bottleneck.
That makes a lot of sense. And you said there was some powerful validation for this whole iron theory coming from studying rare cases.
Yes, this is really compelling evidence. The research team specifically looked at individuals with what's known as mosaic or partial Down syndrome.
Mosaic, what does that mean exactly?
In these rare instances, unlike typical Down syndrome where every cell in the body has that extra chromosome 21,
Right.
in mosaic or partial cases, only a subset of the body cells, or importantly for us, brain cells actually have that extra chromosome. Others are normal.
Okay, so it's like a mix.
Exactly. A mix of cells with trisomy 21 and cells without. And what they found in these individuals was incredibly telling. It served as a kind of living proof of concept.
What did they find?
These patients, the ones with mosaic or partial Down syndrome, had 35% less brain iron compared to those with complete trisomy 21.
35% less iron.
Okay. And crucially, they lived significantly longer. They experienced a later onset and slower progression of cognitive decline compared to those with complete trisomy 21.
Wow, so less of the extra chromosome, less iron, and slower disease progression.
Precisely. It directly reinforces that clear connection we've been discussing. The dosage of the APP gene from chromosome 21, the subsequent buildup of iron, and the accelerated progression of brain damage.
It provides a really strong link between the underlying genetics and the iron pathology.
It does. It's almost like a natural dose-response experiment, making the case for iron's central role even stronger.
Okay, so this iron link is clearly critical in Down syndrome-associated Alzheimer's. But does this have broader implications? Is iron dysregulation an issue beyond this specific context?
Absolutely. And connecting this to the bigger picture is really important. It turns out iron dysregulation isn't an issue confined solely to Down syndrome or even just Alzheimer's. This discovery actually shines a light on a much more widespread issue, one that's deeply intertwined with the broader processes of aging and various diseases.
Okay, like what?
Well, let's look at general Alzheimer's disease first, in the wider population. Elevated brain iron is consistently associated with cognitive decline there too.
Really?
Yes. A large meta-analysis looking at 43 different studies found elevated iron in specific brain regions in 80 patients' areas crucial for memory, thinking, motor control, like the frontal, parietal, and temporal lobes, the amygdala, putamen, cingulate, globus pallidus, caudate.
So many key areas.
Many key areas. And there was also a notable trend towards iron overload in the hippocampus, which is absolutely critical for memory. And what's more, the amount of iron, say, in the caudate nucleus correlated directly with how severe the cognitive decline was for those patients. It wasn't just present. Higher iron meant worse symptoms.
That's a direct link.
It is. Now, there's an interesting sort of iron paradox here that can be a bit confusing.
Okay.
While brain iron levels might be elevated in AD patients, sometimes their iron levels in the blood, the serum iron, might actually be reduced.
Oh, how does that work? More in the brain, less in the blood?
It sounds counterintuitive, right? But it's not necessarily a contradiction. It likely points to changes in how the body transports iron and, crucially, how cells export iron. The machinery gets gummed up.
So iron gets trapped inside tissues like the brain, even if blood levels are lower.
That's the idea. Think of it like a plumbing issue again. Maybe low pressure overall, but a specific pipe, like the one leading out of the brain cells, is clogged, causing iron to back up and accumulate inside that area.
Okay, that makes sense. What else points to iron's role?
Another key biomarker is ferritin levels in the cerebrospinal fluid, the CSF, which bathes the brain and spinal cord. Ferritin is the protein that stores iron inside cells. Studies show that higher levels of CSF ferritin predict worse cognition. They also predict an increased likelihood of someone converting from mild cognitive impairment, or MCI, which is often a precursor stage, to full Alzheimer's disease.
So it's a predictive marker of progression.
Yes, and these higher CSF ferritin levels are also associated with more brain atrophy, more shrinkage over time. It suggests that tracking CSF ferritin could be really valuable.
Interesting. And you mentioned aging itself.
Yeah, beyond specific diseases, iron dysregulation seems to be a hallmark of the aging process more generally. Senescent cells, these are basically aging cells that have stopped dividing but hang around.
The zombie cells, people sometimes call them.
Yeah, that's one term for them. These senescent cells accumulate large amounts of intracellular iron.
Wow.
It seems their ability to break down and recycle ferritin, that iron storage protein, becomes impaired. So iron just builds up inside them. This contributes to the overall increase of iron we often see in tissues as we age.
Huh. And that links to potential treatments.
Potentially, yes. It suggests a fascinating idea. Maybe iron chelation, removing that excess iron, could specifically target these senescent cells. By doing so, you might reduce oxidative stress not just in the brain, but throughout the body, possibly tackling a fundamental aspect of aging.
That's a big idea. Are there links to other age-related diseases too?
Yes, the connections keep branching out. Iron dysregulation has links to other major age-related health concerns, really underscoring its broad impact.
Like cancer?
Like certain cancers, yes. Elevated iron biomarkers, things doctors measure in blood tests, are associated with increased risk and worse progression for various cancers, particularly liver and breast cancer, seem to show strong links.
Why would that be?
Well, cancer cells are rapidly dividing, and they notoriously require high iron levels to fuel that proliferation, that growth. They're kind of iron hungry. Which is why, in lab studies, iron chelators drugs that grab iron have actually shown anti-cancer activity. The idea is to starve the cancer cells of this crucial nutrient. It really highlights the delicate balance of iron too much, or maybe even too little in the wrong context, can cause problems.
Right. What about heart disease or diabetes?
Also links there. Epidemiological studies looking at large populations show clear associations between elevated iron biomarkers like serum ferritin again, and an increased risk for things like heart attacks, ischemic stroke, and type 2 diabetes.
So higher iron stores linked to higher risk.
It seems so. And for diabetes, there's growing evidence suggesting that iron overload might actually precede and contribute to the onset of the disease. It might not just be a consequence, but a potential driver.
Wow. So it's not just neurodegeneration. This iron balance seems critical across the board. Cancer, cardiovascular health, metabolic health.
Exactly. It really underscores the critical importance of iron homeostasis, maintaining that precise balance across so many facets of our health and the aging process. Disrupt that balance, and it seems a cascade of problems can follow in many different systems.
Okay, this is all incredibly insightful, but also a bit daunting. The good news, though, is that this research doesn't just explain problems. It points towards solutions, right? What about these iron chelating treatments you've mentioned?
Yes, absolutely. That's the hopeful side of this. So, iron chelators, essentially, are medications specifically designed to bind to excess iron. They grab onto it.
Yeah.
And the goal is either to help remove it from the body or at least to neutralize its damaging chemical reactivity. In the context of brain health, the aim is usually to reduce that pathological iron accumulation in the brain. Or sometimes the goal might be to strengthen the brain's own antioxidant systems, which are often overwhelmed when there's too much reactive iron around. Either way, you're trying to stop that iron-driven oxidative damage.
And you mentioned conservative chelation.
Yes, that's a key concept, especially for chronic conditions like Alzheimer's. Conservative iron chelation aims to reduce that problematic iron buildup, the stuff causing damage in the brain, without significantly depleting the body's overall peripheral iron stores.
Because we still need iron for other things.
Exactly. We absolutely need iron for vital functions like carrying oxygen in our blood, energy production in our cells, lots of things. So it's a very delicate balancing act. Get rid of the bad, excess, mislocalized iron without causing a deficiency of the good, necessary iron.
Right. So what are the options? What chelators are out there or being studied?
Okay, let's walk through a few, maybe starting with some of the older ones and moving towards newer developments. First, there's diferoxamine, or DFO.
DFO, okay.
This is a high-affinity iron chelator, meaning it binds iron very strongly. It's actually approved for treating acute iron toxicity, like if someone accidentally overdoses on iron pills.
Right, for emergencies.
Exactly. Traditionally, it's given as an intramuscular injection into the muscle, but for chronic conditions like Alzheimer's, DFO has some significant challenges.
Such as?
Well, it has poor bioavailability, meaning not much gets into the system effectively. It has a short half-life, so it doesn't stick around long. And crucially, it has very limited ability to cross the blood-brain barrier.
Ah, the brain security system again.
Yep. It's hard to get it to where it needs to be for neurological conditions. Plus, it carries a risk of acute toxicity itself, with side effects like blurred vision, headaches, even seizures sometimes. So while it works for acute poisoning, it's really not ideal for long-term chronic use.
Doesn't sound like it. Any attempts to improve it?
Yes, researchers have been exploring an intranasal formulation, basically, delivering it as a nasal spray.
To bypass the body and go more directly to the brain.
That's the hope. Enhance brain levels, limit systemic exposure. There have been some promising results in animal models for AD and Parkinson's, but they've been somewhat modest and sometimes inconsistent. There's also a question of whether this route mainly redistributes iron within the brain rather than truly decreasing the overall load. So, work in progress there.
Okay. What's next?
Next up is deferoprone. This is also a high-affinity iron chelator, but it has some key advantages over DFO.
Like what?
Critically, it's orally available, you can take it as a pill, and it can effectively cross the blood-brain barrier.
Okay, that sounds much more promising for brain conditions.
It really is. It's already approved for treating iron overload related to a blood disorder called thalassemia. And because of these favorable properties, it's being actively tested in neurodegenerative diseases.
Which ones?
Moderate doses, around 30 milligrams per kilograms per day, have been evaluated in phase 2 clinical trials for early-stage Parkinson's disease, PD, and also for ALS, Lou Gehrig's disease.
And what did those trials show?
Some promising signs. They showed deferriprone could reduce brain iron in affected regions in Parkinson's, like the substantia nigra. In ALS, it decreased iron in the spinal cord. There were also some modest clinical benefits observed, maybe slowing progression slightly.
What about side effects?
Importantly, the side effects were generally reported as mild and tended to resolve when the drug was stopped. This makes its safety profile seem much more acceptable for the kind of chronic, long-term use needed for these diseases.
That's encouraging.
Yes. And interestingly, it seemed to be most effective in Parkinson's patients who had low serum ferritin levels to begin with.
Low iron stores in their blood.
Right. Which suggests that if the body's main iron stores are already low, the drug might be better able to target the problematic iron in the brain instead of just binding to iron elsewhere in the body first.
Interesting nuance.
Okay.
Any newer compounds?
Yes, there's PBT434. This is a newer compound, chemically different, with a moderate affinity for iron. It also has some modest affinity for copper and zinc, other metals that can be involved in brain chemistry and oxidative stress.
Okay. What's special about PBT434?
Its mechanism is quite exciting. It seems to preferentially target labile iron.
Labile iron. What's that?
That's the unbound, free, and most chemically reactive form of iron. It's the form that's really thought to be responsible for driving that oxidative damage. The idea is PBT434 mops up this dangerous labile iron without significantly disrupting the main stable iron stores that the body needs.
So more targeted at the bad stuff.
Exactly. And like deferriprone, it's orally bioavailable and it penetrates the blood-brain barrier, so it ticks those important boxes.
How's it been tested?
It's been tested in animal models for Parkinson's and also for multiple systems atrophy, or MSA, which is another related neurodegenerative disorder. In those models, it showed protection against alpha-synuclein toxicity, that's a problem protein in PD and MSA, and it reduced oxidative stress.
How about in humans?
Phase 1 trials in healthy volunteers showed it was generally well tolerated and safe. It's currently considered a lead clinical candidate, with development particularly focused on MSA right now, but its mechanism suggests potential for other conditions involving iron dysregulation too.
Okay, so there are definitely options being explored. Deferoxamine with challenges, deferriprone looking promising, and PBT434 as a newer targeted approach.
You've laid out the landscape very well, and it really does offer a glimpse into a potential new therapeutic frontier for these tough diseases.
But it's not simple, right? What are the main challenges?
No, definitely not simple. The key challenges and future directions really revolve around finding even safer, more effective, and reliably orally bioavailable forms that can be used for chronic, long-term treatment without causing significant off-target effects or iron deficiency.
That delicate balance again.
Exactly. We're dealing with an incredibly delicate balance because iron is so essential. The goal is always to target the excess or the dysregulated iron, not deplete the necessary iron.
And when would these treatments be most effective?
Well, based on the mechanisms, it's becoming clearer that iron chelation therapy might be most beneficial if used early, perhaps even in those prodromal stages of AD, or for people with mild cognitive impairment, MCI.
To prevent it from getting worse.
Exactly. The idea is to intervene to prevent or slow the conversion to full-blown dementia by slowing that cognitive decline rather than trying to reverse extensive damage that's already occurred. It's more about proactive intervention than reactive treatment.
Makes sense. Are they likely to be used alone?
Probably not, or at least not always. These therapies are unlikely to be a silver bullet on their own for such complex diseases. For sustained, really clinically meaningful benefits, they might need to be combined with other approaches, other neuroprotective drugs, maybe reparative therapies if they become available.
A multi-pronged attack.
Precisely. Think of it as addressing different facets of the disease pathology simultaneously.
And you mentioned moving beyond just chelation.
Yes, ultimately, the goal might not just be pure chelation, which is essentially just removing iron. Given the complex dyshomeostasis we see iron being in the wrong places, handled improperly by cells,
Right.
the real promise might lie in developing therapeutics that improve overall iron regulation. things that help the body manage iron better, transport it correctly, store it safely.
So restoring the balance rather than just removing the excess.
Exactly. That shifts the focus from just removing a problem element to actually restoring a fundamental biological balance. That feels like a much more comprehensive and potentially more effective long-term strategy for brain health and cellular resilience.
That's a really important distinction. And are these ideas actually being tested now? Are there clinical trials people can look into?
Yes, absolutely. And it's incredibly exciting to see this moving from lab research towards patients. There are current clinical trials underway, particularly for deferoprone, reflecting this effort.
Can you give some examples?
Sure. For Alzheimer's disease, there's the 3D study, which stands for deferoprone to delay dementia. For Parkinson's disease, there are studies like SKY and FAIR-PARC-2 looking at deferoprone.
Right.
And for ALS, there's the FAIR-ALS, the second study, also testing deferoprone. These trials are really critical steps, bringing this promising research closer to potentially becoming real treatment options.
That's fantastic news. It shows real momentum.
It really does. It offers new hope for targeted interventions that could potentially fundamentally change the trajectory of these devastating diseases.
Okay, so let's try to bring this all together. What's the key takeaway for you, our listener, from this deep dive into Down syndrome and Alzheimer's? We've uncovered, or rather, the research has uncovered, a really significant and measurable link. Elevated brain iron and the specific cellular damage it causes plays this critical, maybe previously underappreciated role in the accelerated onset and severity of Alzheimer's in people with Down syndrome.
Yeah, it adds a whole new layer to the amyloid story.
Exactly. This new understanding goes beyond just those amyloid plaques we've known about for so long. It points to these crucial underlying cellular processes, like ferroptosis, that iron-driven cell death, and the alarming role of weakened antioxidant defenses.
Right, the GCLM depletion, the glutathione issue.
Yes. But here's the immense promise. By understanding these unique biological vulnerabilities, researchers are now developing targeted therapies like these iron chelators we discussed. And they offer new hope. It means we're moving beyond a kind of one-size-fits-all approach to Alzheimer's and toward more precise, personalized strategies. Strategies that could potentially prevent or significantly slow disease progression, especially for those most at risk, like individuals with Down syndrome. It offers a future where proactive intervention feels truly possible.
It really does. And maybe a final thought to leave people with. This research truly exemplifies how a deeper understanding of specific biological vulnerabilities, even things that seem like small details, like, localized iron overload in specific brain regions, how that can unlock entirely new avenues for treatment.
Right. It encourages us, I think, to continually question, to look beyond the obvious surface pathologies like plaques and tangles, and to recognize that this intricate dance of elements within our bodies, like iron, holds profound secrets to health and disease. It's this kind of detailed scientific inquiry, looking at these specific mechanisms, that's driving us towards more precise and personalized medical solutions. A future where we can address these complex conditions with greater specificity and hopefully much greater success. This deep dive into the Down syndrome-Alzheimer's connection, it's not just about those two conditions in isolation. It's really a powerful reminder that sometimes the most profound insights that help everyone, come from looking very closely at what might initially seem like a unique or specific case, only to find it reveals fundamental, maybe even universal truths about our biology and how things can go wrong.
That's a fantastic point. Look closely at the specific to understand the general. Thank you for joining us on this deep dive into the fascinating and truly vital connection between Down syndrome and Alzheimer's.
My pleasure. For expert fitness advice and personalized plans, visit armygymnastics.com, your go-to resource to reach your goals. Start your journey today at armygymnastics.com.
Thanks for listening. For all things health and fitness, subscribe to our newsletter today. Find out more at armygymnastics.com.